Thank you so much for joining me for this session. I'm very thrilled to introduce you my passion, which is 3D bioprinting. So it's going to be a bit of an introduction because I am assuming that a lot of people don't really know what the technology is yet, right? Probably you must have heard about 3D printing, but how useful it is with biological applications is something that I think that most people don't really know about. And hopefully by the end of the session, I'll be able to convince you the potential of this technology and how useful it is, how transformative it is. So yeah, let's start with what is bioprinting. So I think more than bioprinting, people would be more aware of 3D printing. So something like this, where you have a 3D printer that can melt and reform plastics like PLA or ABS into any shapes that you feed it, right? And it has a lot of applications actually, not just for making everyday objects, but also in medicine, where it can be used to make implants or guides for skin regrowth, right? You also have very large-scale 3D printers that are used to print houses and other buildings, and also in a lot of creative fields like fashion, where 3D printing is used. But 3D bioprinting is where we are doing 3D printing with human tissue, our own human cells, using living cells and biomaterials. So the video that you see here, it's not a heart, even though it looks like it. It's actually the smallest unit of the human lung, called an alveolus. Um, this has a pointer? Yeah. Yeah. So this is where air is coming in, into this clear area. And you have input of artificial blood. And what is happening is the researchers are seeing whether the air that is being fed inside is able to take blood without oxygen, and by the time it comes out, does it have oxygen in it? The same way that we take in air and get oxygen from air and release carbon dioxide in our body. Although this is interesting and for me impressive to look at, what are the uses of a technology like this, right? And why do we even want to print with cells? If you were to look at cells in a laboratory, which is where most human cell research happens, mammalian cell culture happens, it is done in these flat plates called Petri plates, or in larger versions called flasks. And what happens is that there, the researchers will place the cells on these plates, and the cells will have contacts with each other here. Whatever nutrients you are feeding, it will be a very uniform distribution of it. And oxygen also, there will be a uniform distribution. But this is not how it is naturally, right? How we see cells are in our own bodies. In a 3D cell culture method, like 3D bioprinting, the cells behave like they do in our own body because we are able to trick them to think that they are in our body because of the way that we place them. The cells will have interactions with each other and also with the material on the outside, just like they do in our body. And it is useful to study cells this way because, before that. Because of that, we take polymers such as gelatin, which is something you might be more used to, but there are also synthetic varieties like pluronic, or some other natural derived polymers such as alginate and gelatin gum. We take these polymers, we put in chemicals that cells recognize and will make them behave the way they do in our body, and then we mix them with cells, and instead of using plastic for 3D printing, we make a bioink which we print with for the cell printing. But why do we do this? What are the uses of this technology? Why do we even have to care about this, right? The main established uses of 3D bioprinting would be for drug development and for making disease models. And a few more alternatives that are in the works are for making meat alternatives. For example, lab-grown meat and making it behave the way that real meat does. And also in organ transplants. And now we'll go into a little bit more of examples of things that have been achieved in all of these domains. This is very useful in drug development because if you look at something like basic research, you might get 10,000 different compounds that can solve the needs of a single disease, say, that can be used to treat a single disease. But when you do drug discovery and then you do preclinical trials and it slowly gets whittled down, as soon as it comes into clinical trials, you will see that out of the thousands of drugs, after maybe 5 to 6 years of clinical trials, there will only be one FDA approved medicine or a governmental body approved medicine. And that's because a lot of these preclinical trials, drug discovery and basic research happens first in cells in a 2D environment like this, meaning when it comes to humans, it's not going to behave the same way that it does in these discovery phases, right? And on top of that, these preclinical trials are done in animal models, which has its own ethical concerns and may not behave the same way a human does. Like, you would not expect a mouse, which only shares 40% of its genes with humans, to react to a drug the same way that a human would. But because we cannot immediately go from this phase to these phases, we have to whittle down these drugs somehow. We use a very inefficient way to get to humans, right? But something like that can be circumvented if we were to take our own human cells out of the body, print them into human tissue, and then use those for testing our drugs. And this is something that has already been commercialized. For example, these are the bioprinted skin models that the R&D team at L'Oreal have been working on. So L'Oreal has actually been working on skin models since the 1980s to 1990s. But recently, they have also developed these bioprinted skin models on which they plan to do testing instead of animal testing. And what is exciting about using bioprinting for these preclinical models is that it can be personalized from person to person. Two people can have the same kind of cancer, but in their body, the cancers might not behave the same way, right? Each of our genetic makeups are different, each of our bodies are different, and we can easily predict. We can't use a mouse to predict whether a drug would work in a particular way for one person versus another person. But if we were to get the cells from each person and then print with those, we will be able to not only screen against individual differences within the same disease. If you are somebody who has a very rare disease, somebody might have issues with their mitochondria, somebody might be born with a single mutation in their body that's made their liver not work. Developing animal models for testing drugs against those will be much more difficult than actually using the cells from the same patient and testing drugs against them. An example of this is actually a study from South Korea, where doctors have done surgical resection. I hope it's visible. They have taken biopsies of cancer cells. They have made small mini versions of those cancer tumors. And basically, they tried to mimic the way the blood flows through a blood vessel and the cancer drugs reach this cancer. And then they replicated that with a bioprinted model. And when they tested different combinations of drugs for different people's bioprinted models reacted different ways to the drugs. And the same combination of drugs were able to treat the patients themselves, which is a huge step forward in personalized medicine. Okay. Yeah. And coming to meat alternatives, these are all videos from an Israeli company called Redefine Meat. And they have used bioprinting in 2 different ways to make meat alternatives. So this is one way where they've used a chickpea and soya bean-based paste with different flavor additives to make a beef steak replacement, which looks like this when it is printed and then cooked. And this is another way where they have actually taken cells themselves from the cow and then printed that and matured that to make steaks. So basically, it will taste like the real thing. It will be made of what the real thing is made of, but without any of the ethical concerns for the animal. And the other good part, other than the ethical concerns, would be that it takes much less energy than conventional meats does, like beef, sheep, pork, and poultry. The emissions are much lower for cultured meat. The land use and the water use is also much lower for 3D printed and cultured meat than it is for other meats that we normally consume. And finally, coming to something like organ transplantation, of course, it's going to be very difficult to print an organ because of the complexity of the human body. But the simpler organs like a cornea, which is the outer cover of your eye, right, that focuses light, something like the ear pinna, something like the nose, and skin grafts have already been in the works for a while. So this is a publication which talks about 3D printing, 3D bioprinting a cornea. They print the corneas into these molds, they stabilize it, and then this is what it looks like. And now it's in clinical trials for transplantations. When we want to 3D bioprint something, and now we're going to get a little bit into the technical side, what are the goals that we want to achieve? Obviously, if we are going to print cells, right, we want them to behave like they would in our body. We want them to work exactly the same way. If I want to print skin, say, this is what a small section of skin looks like in the human body. We want them to have the all of the fat deposits, the different types of cells, the blood vessels, the muscles, everything. So that if I were to print it and slap it onto someone who has maybe like a burn wound or a skin excision wound, then it would take over the part of the missing part of their body, right? And then start behaving the same way. But as an engineering problem, it also has to be simple to engineer so that we can make these in a very good amount of time, a reasonable amount of time. We want to balance biological accuracy with design. For example, in the skin, you have these certain folds. Just engineering these folds with a bioprinter can take quite a bit of planning, right? And this is a paper from IIT Delhi and from Saurabh Ghosh's lab where they did that. So there is always a trade-off that we are looking at. Yeah. So there's always a trade-off that we look at. If you have more complex designs, it will behave like it does in the body. But it'll be much harder to produce and even harder to scale larger and larger, right? But if you have simpler designs, it is easy to design and easier to scale, but it might not behave the same way it does in our body. So the role of a bioengineer, the role of somebody who's going to do bioprinting would be to find the best trade-off between these two so that we can make it as simple as possible, but it still works the way it does in our body. And that is what a lot of bioprinting research is geared towards. That is what a lot of bioprinting products will be geared towards. So now I'll move on to some demonstrations and examples where bioprinting has been used. And for this, I'd like to call one person from the audience to help me with the machine. So just one person, please. Don't make me point and call you out, huh, please. What's your name?
Tres.
Okay, all right. Can I know what your background is?
Yeah, I'm a mechanical engineer from Fab Lab Kerala.
Okay, yeah, nice to have you. So you must be used to 3D printing, right? We're just going to use Prontoface for this, actually. So you must be familiar with Prontoface. We're just going to use that. So I'll just show you what the basic components of this 3D bioprinter is. First, we have our compressor here, right here. And I will be switching it on now. It's slowly filling with air. So this bioprinter is operated pneumatically. We have a little input here, which is going to push air out. So I'll just give you a small demo. So now we have our material here that we are going to print with. This is a synthetic polymer. It's fully constructed in the laboratory. So please, just attach this with this input. Yeah. Yep, perfect. One second. And lastly, we are going to add a small dispensing tip. Screw it there tightly. Perfect. Now you can put it inside. And we will be printing something very basic onto this Petri plate. Oh, yeah. Yeah, I'll do that. So this has already been homed, but yeah, you can come here, maybe. Yeah. So I think you're familiar with this interface, right? Yeah. Can you just lift it up by 10, the Z axis? Z. Yeah. Just once more. Yeah. So how this works is that air is going to be pushed through this tube and it's going to hit the little piston here. And because of this air column and the fact that air is being pushed down into it, it is going to go through this needle, the material that we have. And this material normally, right now, we do not have cells because we're not in the lab. But normally, any human cells you have will be mixed into this material. And it's a soft hydrogel, meaning that it's a gel that's made with a lot of water content in it. And that is the kind of environment that cells will thrive in. So, yeah, if you can just extrude a little bit and show us how it's done. Oh, 1 second, I'll set the pressure. Yeah, please. I hope it's visible. Yeah, there's a thin line of material that's being pushed out now. So let's clean that up. Now we'll be doing something called centering, which is basically, oh, sorry. Yeah, not very used to using the machine from this side of it. Huh. Now, you'll just have to bring this needle to the middle point here. Okay. Yeah, it seems okay. Can you just bring it down a little bit further? Yeah. A little bit towards the Y and maybe the X. Little. Yeah. Yeah, and X also. Left. Great. Yeah. Now I'll just reduce the Z height a little bit so that the needle is just touching the bed. Yeah, I'd say this looks good. Now we will load our file, and I have already prepared the files we'll print with. Yep. Now let's just print and it should start moving. Oh, 1 second. All right. Yep, there we go. It's working. Slight adjustment to the pressure, and there we have it. So what we're doing is we're printing this very simple two-layer grid structure. Yeah, thank you for your help. So we are printing with hydrogels, and if you want to see the printing happening, you're free to come closer because I'm sure that you can't really see it from the back, especially if you're a little further back. So, yeah. So this is how a normal basic bioprinting workflow would go, especially from a hospital to a lab. We will have a 3D design that we want to print. It might be something as simple as a little grid structure here, because you just want the cells to be in a 3D environment, and that's it. Or it could be something much more complicated, something like this ear pinna model that you can see here. I'll just pass these models to you guys, and maybe you can take a closer look. Just be careful while handling them, and please pass once you're done. Yeah. This 3D design structure can either be from medical scans, where you have MRIs or CTs or X-rays of the brain, the heart, the lungs. Then you can convert them into a 3D model file. You can then do something called slicing and get the instructions for the printer to print it. You will also be simultaneously making the bio ink. You will then give the printer the instructions to print. And then once you get what you want to print, you'll check, are my cells alive with cell viability assays? And are my cells behaving the way that they're supposed to with something called histology, which is something that's very commonly done in medicine. So I wanted to give you an example of something, a very good paper that came out that actually used a very simple method to get very good results. So this is actually an artificially printed blood vessel that was done by, I think, Harvard Medical University, where they printed this tubule. And inside this tubule itself, the bio ink that is used was a nanomaterial combined with muscle cells that you find in our blood vessels. And this is the construct that they got. Yeah. And this is just a very fancy stained image where the endothelial cells, that is the cells that line your blood vessels, are stained in red. And then the muscle cells are stained in green. So this is what it looks like to the naked eye. And this is what the cells look like. They were able to show that they were able to make leak-proof, stable constructs. And moreover, they actually tested it by passing blood through them and then stimulating the blood to clot. And they found that, yeah, and they were able to find that just compared to just an empty blood vessel, the blood vessel that they had printed with both the endothelial cells, that is the cells lining our blood vessels, and the blood vessel muscle cells, showed much lesser clotting. So not only were they able to make a construct, a blood vessel that could be transplanted for humans, in the case of heart failure, in the case of clogged blood vessels, they were able to show that they made an improved blood vessel where the chances of clotting in the future would also be lesser, right? Now I'd like to call maybe one more person for another print. I think this one should be done. Yep, it's done. One more participant, please, or I can go ahead with it myself. It's all right, I'll go ahead. Hello. Yeah. So what we're doing is printing a small tubule, the same way that we saw in that paper previously that I introduced you. Yep. So here we go. It's not going to be as long because we don't have that much time. But basically, I just wanted to show you with a natural polymer this time instead of the synthetic polymer we used previously, how something like this can be done very easily, even with a very simple printer like this. Yeah. And while this prints, we'll move on to the next examples, which is embedded printing. Yeah. All right. I think I will have to cut this short in the interest of time. Yep. So let's pause the print and let's take this and see. So right now, I hope everybody can see. It is a soft hydrogel. I will show you what it behaves like when I push it out. Like so, extremely soft. When I touch it, it completely disintegrates in my hand. And something like this can be stabilized with some calcium chloride. So this is just a spray bottle of calcium chloride, and I'm just going to spray it a couple of times. There we go. And the little tubule that I've printed, I'm not able to handle it freely. Okay. And I'll just pass this around. You can feel free to touch it, no issues. Yep. So now we will move on to embedded printing. Yep. So we can make very simple tubules, we can make very simple grids, etc., with a machine like this. But how do we try to capture the complex geometries in our body? For example, this is a 3D scan of the aorta, which is the blood vessel, the major blood vessel that takes oxygenated blood from our heart to the rest of the body. When the 3D printer prints layer by layer, it can go 1 layer, 2 layer, 3 layers, like this, like this, like this, so on. But what if it comes to this part, where it's going layer by layer? How can it print something in midair? It can't. If you were to try to deposit the material, it would just drop down. And the same with all of these very beautiful but complicated structures that we regularly see in biology. In regular 3D printing, you would do something called using supports. This is actually a femur, which is the bone in our knee, right? Bone in, yeah. And you use these things called supports, where you print these little structures that you can later take away and then smoothen the surface of the print, right? But how would you use supports in a hydrogel system? You will not be able to, which is why there is a technique called FRESH, or free-form reversible embedding of suspended hydrogels, that in which you take the bio ink and then you print it into a reversible support bath. And I'll also be showing that to you today. So I'll just set that up quickly for you right now. So this is the support bath that we'll be using. It is a synthetic polymer again called pluronic. I'm so sorry. I really wanted to show you a print, but we're having some technical difficulties with the printer. But I can show you some examples of what the fresh print would have looked like. It's basically a suspended kind of printing. I'll pass around the samples for you, and you can take a closer look. Yeah. All right. Yeah. Yeah. So fresh printing was a method that was introduced in 2015 by the Carnegie Mellon University's bioengineering department, where they have a gelatin bath where you can actually print a lot of different kinds of bioinks in a suspended format. It uses something called a Bingham plastic liquid. And this is sort of a closer view of the suspended bioink in the material itself. And this is a small model of the human heart they were able to print. And all of these green little flecks you see are actually live human cells. And with this, it's quite exciting because a Chinese group was able to print a small artificial liver model, which is definitely not as complicated as the real human liver, right? But again, when we come to that trade-off, they were able to show that even with a very simple engineered model of just these primary mouse liver cells and blood vessel cells, they were able to take the printed construct, transplant it into the mouse, and then all of these green little wiggles you see are actually blood vessels that the mouse's body grew into the transplant. So basically, it's an indication that the transplant was accepted into the mouse, and it started behaving similar to how a liver would in the mouse, which is a very exciting step towards actually transplanting and printing and transplanting organs with the 3D bioprinting. And similarly, with this same technique, people were able to print vascular networks where they found healthy blood vessel cells. And this can be also used for better drug testing before these drugs come to humans for human clinical trials. So, yeah. That's it for now. I'm really sorry about the technical difficulties with the machine. But we can definitely discuss now if anybody has any questions. Please. I'll just pass around the small sample. So I had already prepared a sample of the what I wanted to print for you before, just to show what it looks like when it's taken out of the printing liquid. So, yeah. You can feel free to touch it, but don't handle it very roughly. It's still a very delicate biomaterial. So, any questions about the technology? Yeah, please.
You told about the suspended printing.
Yes.
If you are printing a closed body
Yes.
and the gel will be trapped inside, right?
Oh, that's a very good question actually. So the way this gel works is that it is a gel that is thermo-reversible, meaning if I were to keep it in cold water, which is what I've done here actually, this is what I took that little lung model out of. You can see that it is completely liquid, right? And that's because I kept it in ice water here in preparation. And this is the same gel when it's solid. So as long as you leave a very small gap for the gel to drain out when it shifts into the liquid form, you should be able to wash it out quickly. So yeah, that's how it's used. Any other questions?
I'm assuming you used sodium alginate for the second.
Yeah,
one.
exactly. Sodium
So
alginate.
it's not about the technology. I want to
Yeah.
know, since you sprayed calcium chloride,
Yeah.
it will release salt, right?
Yes. Yeah, you could say that.
So if you are using cells,
Mhm.
I'm just asking,
Yeah.
will it not affect the viability of the cells?
That's a great question, actually, which is where the bioink optimization comes in. So the concentration that I've used for demonstration purposes here is 100 millimolar of calcium chloride. Oh, and I'm sorry, the second one is actually a polymer called gel and gum, not alginate. The third print which I showed with the fresh, that is alginate. So yeah, gel and gum, you can cross-link it from anywhere like zero point 0.1 millimolar all the way up to 1 molar. So you just have to find the balance of salt ions at which your cell's viability are not affected. Yeah. So if you wanted to use something like skin cells, right, and print with skin cells, they are very calcium sensitive. But so I would suggest that you don't go with calcium at all. You go with something else like strontium or barium. But if you were to print with fibroblasts, it doesn't really matter that much. Yeah. So as per application.
And you said that you have these three materials, right? First
Yeah.
is the to make the bioink
Yes. Yeah.
cells, the primary this thing, and another material. Like
Oh yeah. So the cells, the growth factors for the
Okay,
cells,
so growth
and the
factors.
polymer. Yeah. That's it.
Is there any other questions?
Thanks for the wonderful section. I have a question on like general 3D printing when there is so much movements, right? Travel points from one point to another.
Sorry, I'm not able to hear you clearly.
Like you have many travel points in general 3D printing, right? Like it moves many places to make a complex shape or any.
Oh yeah,
structure.
yeah, so, yeah, got it.
So
Huh.
similarly, when you do this in the biomedical, how the structure is not affected or there is no extra extrusion. Like you use pressure to control the amount of.
Ah, yeah, perfect question. Yeah.
Yeah.
So again, it really comes down to the trade-off between biological likeness and engineering simplicity. So there is actually no better way to print these cells than with pressure, because okay, yeah, I hope it's visible. But here you see that there is an empty space here, right? That means that until this piston, only the cells that are in here are in contact with absolutely nothing else. So printing with pressure means that you maintain sterility and you don't put the cells in as much pressure as maybe if you were to drive a screw through it to push it out. But pressure does have a big effect on cells. So anything more than 2 bar of pressure, right? Or yeah, is not good for them and they start dying off. So the material you use has to be such that, and this is hard work, but you have to do it such that the cell viability does not go down. So there's always a trade-off.
Another question is like if there is an extra structure forming because of movements,
Yes.
like leakage or something, right?
Ah,
So
right.
how do you clear this off? Like in general therapy, we do post processing, right?
Mm-hmm.
So similarly, do you have any post processing method or any ideology there?
Yeah. So in 3D printing, the post-processing would be something like sealing and sanding, etc., right? Here, the post-processing is done less by us and more by the cells that we put in there ourselves. So we'll be putting in some cells sometimes that will produce their own biomaterials, just like our cells do in the body. Like our bones are made by our cells. Our fat deposits are made by our cells. What we can do is we can guide our cells in the post-processing stage to behave like they would in our body and patch in any gaps or leakages or do the finishing themselves. So that is the approach that's taken.
Thank you.
Yeah.
Can I know if you have already 3D printed the part and you place it into the body, right?
Yes, yeah.
If it's then the nerve cells will automatically grow, or you should be doing that by
Well,
operation?
the ideal thing is that our body will perfectly accept it 100% and the cells start colonizing it immediately. But in real life, it might not happen exactly the same way. So at least for a little while, you'll probably have to take drugs so that it's not rejected. And you will probably have to do some amount of care medically so that whatever you put inside is not rejected. But what researchers are working towards is that it's like a one and done thing.
One more thing I need to ask: if you are printing a muscle cell or like lungs, there is a diaphragm movement and all. So
Yes.
can you replicate that movement with the 3D printing?
Just with a 3D printed structure, perhaps, because if you were to take something like heart cells, right? If you grow them even in a Petri plate in the right way, they start beating by themselves. they start showing that contractile motion by themselves, even in a Petri plate. So the idea is that you engineer the organ, you print it in such a way that you enable it to behave the way it does in your body, which for the heart might be printing, which for the lung would be being able to expand and contract, like the video that I showed previously. But of course, because of the complexity of the human body, even in very big places like MIT and Harvard, we are still only in the initial stages. But perhaps in the next 10 to 20 years, you will be seeing a lot more breakthroughs and advances. Yeah.
Okay, from my knowledge of like on the bioprinting, I know that like ear bones and small bones are trying to be implanted into the human
Right.
body.
Yeah, yeah.
And what will be your first product like that?
Well, actually, bioprinted corneas are already there in clinical trials. A lot of the bone implants that you see is actually thermoplastics like PLA, ABS, PCL, PEEK. Those are the ones that are printed, and then the bone cells are seeded onto them, and or they are just planted directly into the body, and the body takes over the seeding, colonization, etc. But if you were to look at actual printed cells and what will come to market first, it's probably going to be something like the cornea, something that is on the outside of the body that doesn't require blood vessels or too much complexity.
And you will be focusing on which?
No, we are actually a healthcare startup incubator. This is one of the technologies that we offer. So hopefully you will focus on it once you come to our incubator. Yeah.
Okay, thank you.
Hello, Sai, with your permission.
Yeah.
So just to add to your answer, what this gentleman has asked for.
Yeah.
I'm sorry, I'll come, friend. So we have organized this workshop as a part of the awareness for the technology that we have in our incubation center, which is Dr. Moopen's iNEST, that is BIRAC supported incubation center, which is hosted in Wayanad in Moopen's Medical College. So the intention behind organizing this is that like many of you people would have some kind of concepts wherein like you will have to work upon, and we have the technology with us, scientists, the full laboratory, including the stem cell facilities and all, and a dedicated biofabrication engineer. So provided that like you have a concept, we are happy to support you to develop your concept in our facility. So this is the main intention behind this workshop, and I would also agree that like having a session for 1 hour wouldn't suffice to completely understand the technology. So like we are open to have you there in our facility. You come visit, discuss with our scientists. We have around 6 PhD holders, PhD postdoc holders, and you can have a discussion with them. Then maybe you can mature your concept by discussing, and then take up your concept to fruition. So this is what like we wanted, and this is just a kind of a glimpse of like what we can show to you in this day. Yeah. Thank you, Sai.
Thank you. No other questions? Okay, yeah, I think we can wrap up then. Thank you so much for your attention. all
Thank
right?
you so much.
Yeah.
We will also be leaving our, okay, yeah. Dr. Moopen's iNEST, you can just Google out and then reach us. And we also have a small standee outside, maybe like we can scan and register so that like we also be able to get back to you and initiate the communications. So thank you so much for attending this workshop. Hoping that like at least few of you would be reaching out to us. Thank you.